The nucleus is the largest structure in the cell. It's also the cell's control center. A cell typically has one nucleus, but there are cells that don't follow the rules. An erythrocyte, red blood cell, when it's mature and functioning, it's ejected its nucleus. I'm done with you, man. Get out of here. Okay? Skeletal muscle has many nuclei. Now, there are other cells that will have 2 or 3, but sometimes even that one nucleus is determined, the shape of that nucleus is determined by the shape of the cell. So a long, slender cell will typically have a long, slender nucleus. A short, boxy cell typically has a more rounded nucleus. However, there are some cells, like our white blood cells, that have a multi-lobed nucleus or a horseshoe-shaped or a great big, huge round nucleus that takes up most of the cell. So, important thing to remember here, the largest structure, the control center, and the number of nuclei vary, as well as the shape of the nucleus based on the cell. The nucleus is enclosed in a double membrane that's called the nuclear envelope or the nuclear membrane. It separates the cytoplasm from the neoplasm, which is the fluid within the nucleus. Each of the 2 layers of that nuclear envelope are phospholipid bilayer, just like we've seen with the plasma membrane. Externally, it's continuous with the rough ER. There are holes called nuclear pores. They're open passageways that are formed by proteins that extend through the nuclear envelope. So the pores are larger, though, than ion channels, and they allow for the passage of larger molecules in and out of the nucleus. So proteins can come into the nucleus and out of the nucleus, primarily messenger RNA. However, we do have ions and smaller water-soluble molecules that will pass through the nuclear pores as well. The nucleus has a dark staining little round structure within, and that is called the nucleolus. It's composed of protein and RNA, and that's where we make those subunits of the ribosomes that we saw in a previous video. Now, chromatin is DNA. So when our cells are not dividing and making new cells, the DNA and all the proteins that are a part of the contents of the nucleus are in the form of just like a thread. We call it chromatin. And really it just looks like an unrolled spool of thread or yarn that you just pulled and pulled and pulled, and you've just got it all piled up. So chromatin only becomes tightly coiled chromosomes when the cell is dividing. Deoxyribonucleic acid is what we commonly just call DNA. It's located within the nucleus, and then, of course, as we mentioned previously, there's also a little bit of mitochondrial DNA. So DNA is made of repeated monomers of deoxyribonucleotides. If you remember back when we talked about the macromolecules, and we mentioned that nucleotides are the monomers of the big polymers of DNA and RNA. Each of the 4 nucleotides that make up DNA contain a deoxyribose sugar, a phosphate group, and one of 4 nitrogen-containing bases. Now remember, we talked about this previously. The nitrogen-containing bases are adenine, guanine, cytosine, and thymine in DNA. Now, we hook those nucleotides together with a phosphodiester bond between the phosphate parts of those, and that creates one strand of A's, T's, C's, and G's. So one strand of DNA. But DNA exists as 2 complementary strands. So we have 2 strands of nucleotides, A's, T's, and C's, and G's. Okay? A's on one strand will bind to T's on the other strand. C's on one will bind with G on the other. And that is the only way these bases will interact with each other. So A's form a hydrogen bond with T's, and C's form a hydrogen bond with G's, and that gives us the double-stranded DNA that makes up the 46 chromosomes that humans have. A human somatic cell, just the normal body cells, not red blood cells, not sperm and egg cells, each have 46 double-stranded DNA molecules composed of over 3 billion pair of nucleotides. To help package that DNA molecules within the nucleus, each of those long double helix strands will wind around proteins called histones. When they do so, that forms something called a nucleosome. Now, when the cell is not dividing, as we mentioned a while ago, DNA and those proteins are just in the form of a long stringy mess. We call that chromatin. When the cell is getting ready to divide, the DNA chromatin, long stringy, wraps around the proteins, the histones, and create tightly coiled masses called chromosomes. But again, when the cell is not dividing, long stringy mess. When it is dividing, tightly coiled, wrapped around the histones to form chromosomes. Functionally, DNA is arranged into smaller little pieces called genes that are segments of nucleotides that provide the instructions for making a particular protein. The average length of a gene is about 3,000 nucleotide base pairs. Associated with each gene is a promoter region that's sort of like a start button when we start using that information to make a protein. In addition to that, there's a terminal region that's like a stop button that stops the copying of the information into an RNA molecule, and then that RNA molecule is used to make the protein. So you're probably familiar with chromosome. Fewer people have ever heard of chromatin, but our genetic DNA is typically arranged as chromatin, and that looser arrangement, not being all tightly coiled together, allows the cellular structures better access to the genes so that they can make proteins from that gene. So I think of your DNA very much like a cookbook, and each individual gene is a recipe that tells us what to put together and how to put it together in order to make a product. The DNA within the nucleus directs protein synthesis. Now remember, synthesis is making, and protein synthesis occurs at the ribosomes in the cytosol. So there are two major events involved in making proteins. Transcription occurs in the nucleus by RNA polymerase enzyme, and that is the forming of an RNA molecule with ribonucleotides using DNA as the template or the recipe. Okay? Translation occurs in the cytoplasm by ribosomes, and that is where that specific protein is made from amino acids by the direction of that RNA molecule that transcribed the DNA information. Now, don't panic. We're going to keep on with this until you get it.